Leaf disc transformation by Agrobacterium tumefaciens. So in the last example, we have studied that how two plasmid strategy can be used to infect leaf tissues or any explant. So in this topic, we are specifically focusing that how some portions of the leaf that may be called as leaf discs, how they can be transformed by using Agrobacterium tumefaciens that contain either single TI plasmid that contain foreign gene or even Agrobacterium that contain two plasmids. And in this case, the leaf discs, they are made from the leaf tissue by punching, and those leaf discs, they are then disinfected so that all the other sources of microbes, they may be eliminated. And the sterilization, it can be performed by using certain chemicals like sodium hypochlorite or ethanol, or some other disinfectants can be used. Once you have carried out the sterilization, then leaf discs, they are placed in a suspension of Agrobacterium, so that now the leaf discs, they contain area that have been damaged all around, and it also contains natural source of phenolic compounds in their wounded tissue that can induce the expression of various genes in the Agrobacterium. So the incubation, it can be performed overnight or even for a longer period of time. So once incubation is completed, then leaf discs, they are placed on a culture medium that contain kanamycin and carbanicillin. So here, kanamycin will determine that whether foreign gene has been inserted or not. So basically it is going to act as a selectable marker. जबकि जो कार्बेनिसिलिन दूसरी एंटीबायोटिक्स है, ये एग्रोबैक्टीरियम की जो रेप्लिकेशन है या मल्टीप्लिकेशन है या उसकी सर्वाइवेबिलिटी जो है उसको इन्फ्लुएंस करती है, यानी कि ये एग्रोबैक्टीरियम की ग्रोथ को इनहिबिट कर देती है ताकि हमारे पास सिर्फ अब वही सेल हैं जो कि माइक्रोबियल फ्री हों, क्योंकि वंस फॉरेन जीन हैज़ बीन इंसर्टेड, एग्रोबैक्टीरियम नो मोर रिक्वायर्ड. Now in the next step, leaf discs, they are being transferred on a culture medium that contain appropriate amount of phytohormones like auxins and cytokines, so that organogenesis is there, plantlets can be produced, and then those plantlets that have been grown under laboratory conditions or in vitro, they can be excised and then they can be transferred to the soil. So again, we are going to focus on the basic strategy that how leaf discs, they can be transformed by using Agrobacterium tumefaciens that contain disarmed TI plasmids. So in this case, the leaf discs, they can be prepared from the leaf tissue and then they can be surface sterilized. After surface sterilization, they are incubated in the suspension of the Agrobacterium tumefaciens, and then leaf discs after incubation, they are transferred, blow dried and transferred on culture medium that contain antibiotics like carbanicillin or kanamycin, so that here it is first transferred for shoot induction, and then it is further transferred on a medium that contain kanamycin and carbanicillin for 20 days, so that plantlets, they can be generated from the transformed cells. After that, when plantlets, they are generated, shoot formation is there, then they are further transferred to the soil as soon as roots appear after 4 to 7 weeks. So this is the basic strategy by which we can transform leaf discs. So we can say that plant biotechnology, it can be used to transfer our gene of interest more rapidly as compared to conventional plant breeding techniques. So it has been used to transfer genes related to insect or herbicide resistance, and even to increase their resistance to drought conditions.